Hello students, welcome to this new session on biological databases. So as we all know that due to the advancement in the field of molecular biology and sequencing technique, huge amount of DNA, RNA and protein sequence data is being generated. But apart from the sequence data, another type of biological data is also being produced at a very rapid pace, like structural data. It may be the structure of RNA molecules or protein molecules which are determined by circular dichroism or X-ray crystallography or NMR. So that kind of structural data also need to be saved in the databases. Apart from that, there are two-dimensional gel electrophoresis data. Then the microarray data is also being generated. So also mass spectroscopy data is being generated. Then the bibliographical data, that is the articles, research articles or the books which are being published in the field of biology, that data also has to be stored in some sort of databases. So in this lecture, we are going to discuss about what is database, what are the different types of databases, that is the flat file format of the database, then relational database and object oriented database. These are the three different types of database management systems. And later on, we will discuss about the biological databases, types of biological databases, that is primary, secondary, and specialized databases. And we will also discuss about some of the examples of these biological databases.
So what is a database? Now, database is a computerized archive in which the data is stored in a structured fashion, which can be from which the information can be retrieved by providing a query. Now, in this structured collection of data, it involves computer hardware as well as software, and you can retrieve the data by variety of ways. And any large collection of information can be established into the database. Then database management. It is the organization and manipulation of data in the database. Then database management system, it's a software package that provide all the functions which are required for database management. And database system, a database together with the database management system is called as database system. So these are some characteristic features of the database. Database is a collection of data. Now, there is a difference in data and information. Actually, data is simply collection of raw facts. And when you process this data, what you get is information. So in the database, usually data is stored, and in the biological databases, we store biological data. That may be nucleotide sequences like DNA and RNA sequences, or it may be a protein sequence database in which we store the amino acid sequence of the protein, or it may be a protein structural database in which we store the three-dimensional structures of the protein. So the basic characteristics of databases are they are structured, means the data is not stored randomly in the database. They have some order. Then the next characteristic is they are searchable. Just like our book has index in the front of in few front pages, so that by using that index we can search the actual content in the book. Similarly, the databases also has index through which we can search the database in a minimum amount of time. Then they are updated periodically. This is very important feature of database, that the existing information, existing data, if some new data is found related to that, it will be immediately updated. As well as databases are cross-reference, means they are hyperlinked. Means a particular database is always linked with other databases also. Just like suppose there is a nucleotide sequence database and that nucleotide sequence is published in some article, you will get the link of that article in that sequence information itself. Or suppose there is a protein sequence file is there in the database, and its structure is also available in the structural database. So you will have a hyperlink in the protein sequence data which will connect you to the protein structural database. So all these databases are cross-linked with each other with the help of hyperlink. The database also include associated tools or softwares necessary for access, updating, information insertion, or information deletion. Now, basically, there are three different types of database management system. The very basic one is flat file system. The second one is a relational database system, and the third one is object-oriented database system.
Now let's see some features of database. Now a database is a computerized archive used to store and organize data in such a way that information can be retrieved easily via a variety of search criteria. So computerized archive means it's a place where we store some important things. So database is a computerized archive. We store some important data into computer. So they are computerized archive which are used for the storage and organization of the data. Means we store the data in a very ordered fashion in the database. Now the storage in the database has one advantage is that we can retrieve the data from the database by providing variety of search criteria. Now these databases, they are composed of computer software and hardware for data management. So the database consists of a data server, which is a master computer with the huge storage capacity with it. And it also has a software component called as database management system. A very good prominent example of it is SQL servers or SQL database management system. So databases consist of two things. One is computer hardware and other is database management system software for the data management. Now the chief objective of the development of database is to organize data in a set of structured record to enable easy retrieval of information. So what you do instead of storing data haphazardly or randomly into single file in the database, we organize data in set of structured records. We divide the data into certain fields or certain entries, and then we store the data into database. Now, if we store this data in such an organized way, the advantage will be at the time of retrieval of the data. You can retrieve the particular piece of information easily from the database. Now, each record that you're going to store in the data is called as an entry, and it should contain a number of fields that holds the actual data items. For example, field for name, phone number, address, or dates. Let's understand it with the analogy of your data storing into the college database server. Now your data entry or your data record, it will be consist of variety of fields or the information about you or the data about you. Like it will have a field for name, then your phone number, your address, your date of admission, your class of admission, or your SID number. This whole lot of data about a single student, it will be called as an entry or a record. And each entry is subdivided into fields. Then for the retrieval of a particular record from the database, a user can specify a particular piece of information called as value to be found in a particular field and expect the computer to retrieve the whole data record. So again, let us understand it with your college database server. Now suppose you want to find out whole information about one student. So we can provide that database server a single piece of information about that student. The piece of information may be SID number, it may be mobile number, it may be class, it may be name, it may be any other field that we stored along with that record. So when you enter only that single field or single piece of information into the database server, it will retrieve the whole information about that student and show you on the screen. And this whole process is called as making a query. You simply provide a piece of information to computer and whole information about that single piece or a single value, it will retrieve from the database and show you on the screen. Now, although data retrieval is main purpose of all databases, the biological databases often have a higher level of requirement known as knowledge discovery, which refers to the identification of connections between the pieces of information that were not known when the information was first entered into the database. So in case of biological databases, they are even more advanced as compared to the simple databases. What it does, it connect all the raw data facts and identify the connections in between them, convert the data into information so that knowledge discovery will be easy for the user. And that's the whole idea of biological databases. They are very helpful for knowledge discovery after analysis of the data. Then types of databases. Now there are three types of databases that are being used. One is flat file format, other one is relational database, and third one is object-oriented database. So originally all databases, they used to same format, that is flat file format, which is a long text file that contains many entries separated by delimiter, a special character such as vertical bar. Within each entry are the number of fields separated by tabs or commas. So it's a very simple type of database file. In a single file, your entire data can be entered, and each of the entry is separated by the delimiter, which could be a vertical bar. Now, the entire text file does not contain any hidden instruction for the computer to search for the specific information or to create reports based on certain fields from each record. Thus, to search a flat file for the particular piece of information, a computer has to read through the entire file, and obviously an efficient process. So it would be just like a book without index. Just imagine a book without index. Now, if you want to search something from such a book without index, you have to go through all the pages of that book. But if the book has an index, then you can search the topic of your interest in the index, and then you can directly go to that particular page. So flat file format databases are just like the books without index. Now, this is manageable for small databases. Flat file format can be used for very small databases, because if you use flat file format for the large databases, then computer will take a lot of time for the searching process. But a database size increases or data type become more complex, this flat file format of the database can become very difficult for information retrieval. The computer will take a lot of time to find the information from the complex data or the large size of the databases if you're using flat file format. Indeed, searches through such a file often causes crashes of the entire computer system because of memory intensive nature of the operation, and sophisticated computer software programs for organizing, searching, and accessing data have been developed, and they're called as database management systems. So earlier, in the earlier days, this flat file format system of database was used where whole data is stored in a single file and each entry is separated by the delimiter like vertical bar. But nowadays, as the amount of data is also increasing, as well as the complexity of the data is also increasing, for many of the databases, we are not using flat file format, but instead of that, some database management systems have been developed. So these systems contain not only raw data records, but also operational instructions to help identify hidden connections among the data records. Now, the purpose of establishing a data structure is to easy execution of searches and to combine different records to form final search reports. They are classified into two types. One is a relational database management system and object-oriented database management system. Consequently, database employing these management systems, they are known as relational databases or object-oriented databases. So these two types of database management system, that is relational and object-oriented, they can handle a lot more complex data as well as large size of the data, and we can provide variety of functions in this relational database management system or object-oriented database management systems, so that the hidden connections among the data records can also be identified with the help, which is not possible in case of flat file format of the database. So let's understand how these all three different types of databases management system works. That is flat file, relational database, and object-oriented database. So let's start with the relational databases. As in flat file format, we were using a single file. Here in relational databases, instead of using a single table as in flat file format databases, a relational database uses a set of tables to organize data, and each table also called as a relation. So you organize the data into different tables, and you establish the relationship among the tables which contain data, and that's why this is called as a relational database. Now, in each table, there are maybe two or more columns are present. Now, these columns, they represent individual fields, and rows represent values in the field of the records. The columns in the table, they are indexed according to the common feature, which is called as an attribute. So, if by this common feature or by this common value, all those tables can be cross-referenced, and it is made up of these columns and rows. To execute a query in the relational database, the system selects linked data items from different tables and combines the information into one report. So whenever you make a query to a relational database, what system will do? System will find out the linkage among the data records, and all the linked data records, they will be provided to the user in the form of single report of information, and therefore specific information can be found more quickly from the relational databases than from the flat file format of the databases. Now, relational databases can be created using special programming language called as structured query language, or the short form of it is SQL. So in this slide, you can see the example of a flat file format of the database. In the box, some information about the student is recorded. Now, each student information is called as an entry, and each of the data record in that entry will be called as a field. So there is a field for name, state, course number, and course name. Now at the end of every entry, it will be separated from the next entry by the delimiter. It is represented by the vertical bar. So when one entry ends, you simply place the vertical bar, and computer will recognize it as the end of the entry, and a new entry will be started. So you can see John Smith is the name of the student. The state name is Texas. The course number is Biol 689, and the course name is Bioinformatics. So in this way, the information of these all students is given in the single file called as flat file format. So simple example of student course information expressed in the flat file, which contain record of 5 students from 4 different states, each taking a different course. Now each data record is separated by vertical bar. So once the information of one student finish, then you place the vertical bar. So it indicates that the entry has been finished here. So it contains 4 fields describing name, state, course number, and title. Now the same information of the students when we store in a relational database. Now you know that in the relational database we store the information in the different tables. The tables may consist of 2 or more columns, and each column represent a field, right? And each row represent the entry or the information for that particular field. So you can see there are 3 tables: table A, table B, and table C. Now in the table A, the numbers are assigned to the student. You can see students number 1, 2, 3, 4, and 5. Then names of the students are given in one column. So that is one field for the name, in which all the students' name have been entered. Then there is another field for the state. You can see the state name has been entered for that particular student. Now in the relational database, at least one column will be common among 2 tables. Like in table A and table B, you can see the student number column is common. The student number column is present in table A as well as in table B. Then in table B and table C, you can see the course number column is common. That is, course number column is present in table B as well as in table C. So by using these common columns or common fields, the relation in between the tables can be established, and it will be very easy for searching the particular piece of information or particular data from this relational database. So if one has to ask a question, which course are the students from Texas taking? So the database will first find a field of state, which is present in the table number A. So you can see that the state field is present in the table number A, and it will look up for the Texas. So here you can see there are two students. They are from the state of Texas. Now the question which we ask to the database is that which courses are students from the Texas are taking. So we ultimately want to find out the course name, which courses are being selected by the students from the Texas state. Now when you find out in the table number A that the student number 1 and student number 5, student number 1 and student number 5, they are from state of Texas. So this will return student 1 and 5 as a result. Now the student number are co-listed in the table B. You can see here that the student number are co-listed in table number B. That is, student number column is present in table A as well as in table B it is present. Now in table B, you come to know that student 1 and student 5, they opt for Biol 6 at 9 and Math 172, right? Now you can see the course number column is co-listed in the table number C. You can see course number column is common. Course number column is present in table B as well as in table C. So again, you will see, or the database will find out that this Biol 6 at 9 is actually bioinformatics, and Math 172 is actually calculus. So the end result which will be shown by the database will be bioinformatics and calculus. So the final report that you will see on the screen, uh, it will show you that the students from the Texas, they are taking courses bioinformatics and calculus. So instead of searching for the whole file in the relational database, it find out the relationship among the tables, and by cross-linking the particular field with the another table, it will find out the information. So that is very easy as compared to the flat file format where the computer has to search through entire file. Then next is object-oriented databases. Now one of the problem with relational databases is that tables used do not describe the complex hierarchical relationship between the data items. Now in order to overcome that problem, object-oriented databases have been developed that store data as objects. So in an object-oriented programming language, an object can be considered as a unit that combines data and mathematical routines that act on the data. Now the database here, it is structured such that objects are linked by the set of pointers. Now, so instead of putting the data in the form of table as in the relational database, in the object-oriented database, we represent data in the form of object. And to link the different objects, we set the pointers in between those objects. So these pointers, they are defining predetermined relationship between the objects. Now searching the database involves navigating through the objects with the aid of the pointer, linking different objects. And for object-oriented database development, we can use programming languages like C++. So this is the example of object-oriented database. Now each of the field, now here fields are state, student name, course number, course title, these are all your fields for the particular entry, right? Now each of the field, each of the record, it will be sorry, not record, only field. Each of the field, it will be identified as a separate object. So there will be object for the state, there will be object for the student, and there will be object for the course. Now there are five states. You can see in the state objects there are four states: Kansas, Illinois, New York, and Texas. Then in case of student objects, there are five students. They will be assigned with the student name and student number. And there is a course object where uh, course number and course name, they are assigned. Now, in order to establish the relationship among these objects, the pointers are given. So each object is connected with the pointer. Like in case of uh, state object Texas, the Texas state object is connected with the student object by the pointer. You can see this pointer, it is connected with student number 5, and again this pointer is connected with student number 1. So it indicates that these two students are from state of the Texas. Then this particular student object is connected with the course object, where we can see that the student number 5 is taking the mathematics course math 172, that is calculus, and student number 1, it is taking the course bio 689, that is bioinformatics. So in this way, the objects are connected by the pointers to establish the relationship or connection in between the different objects. So the above students course information can be used to construct an object-oriented database. There will be three different objects: student object, course object, and state object. And their interrelations are indicated by lines with the arrows. So to answer the same question, which course are students from Texas are taking, one simply need to start from the Texas in the state object, which has a pointer that will lead to the student number 1 and student number 5 in the student object, and further pointer in the student object point to the course each of the two students are taking. Therefore, a simple navigation through the link object will provide you with the final report in the object-oriented database.
So why there is a requirement of biological databases? So as we have already discussed that enormous amount of sequence and structural data is being determined on the daily basis. So there is exponential growth in biological data. And if we try to write whole sequence information or sequence data of nucleotides, the paper in the world will not be sufficient to write that sequence data. And that's why we rely on biological databases, which are computerized databases and can store enormous amount of data. Now, data in case of biology, it could be anything, like it may be a genome sequence, it may be a three-dimensional structure of the protein, or it may be a simply 2D gel analysis, or it may be a mass spectroscopy analysis data, it may be a microarray data. So there are no longer published, which are no longer published in conventional manner, but directly submitted to the database. And a very important thing in today's scientific world is that if you have some sequence data or structural data, you have to communicate it with database. They will provide you with the accession number, and only after that you can publish that data in your research article. So submission of data to the databases is precondition today, then essential for biological research. So, as we know that knowledge increases by sharing it. So, if your data is submitted into the database, it is available to other researchers and they can enhance that knowledge further. So, it is very essential tool for biological research, for submission of data as well as for retrieval or access of the data. The only way to publish massive amount of data without using all the paper in the world is biological database. So distribution of sequences. Initially, if you see in 1968 to 1985, people were publishing the sequences in books and articles. Later on, computer tapes were available, and people use computer tapes since 1982 to 1992 for publication of the sequences or distribution of the sequences. From 1985 to 1990, floppy disk, 1989 CD-ROM, 1989 FTP, that is file transfer protocol. From 1982 to 1994, it was online services. Since 1993, we are using www, that is World Wide Web, and since 2001, we are also using DVDs for distribution of the sequence data. So these are some statistics about the biological databases. Today in the world, there are more than 1,000 different biological databases are present, and their size is very uh, wide. Like few databases, their size is less than 100 KB, and there are a few databases whose size is greater than 20 GB. Like there are some DNA sequence databases whose size is more than 20 GB. There are some protein sequence databases whose size is around 1 GB. 3D structure databases 5 GB or more. Other databases may be even smaller. So these databases, they are updated frequently. Now the frequency of the updation is very different. Some databases, they are updated on the daily basis, some are on annually, and some databases, they got never updated. So many of the majority of the databases, they're accessible through the web. Like you just connect to the internet, you type in the URL address of that database and you can access it. So some databases are free, but some charge you for their access. So this is a list of some databases in the field of molecular biology. There are around thousand databases in this field. So categories of databases for life sciences. Now there are different dedicated databases for particular kind of data. For example, sequence databases. So there are databases for nucleotide sequences like there is GenBank, DDBJ, EMBL, and there are databases for proteins like SwissProt, Trembel, or UniProt. Then there are databases for genomic sequences, mutation, polymorphism, protein domain, family, proteomics, 3D structure, metabolic networks, regulatory networks, or bibliography, where you can find out the articles or books. Or there are specialized databases for expression, like the microarray database. Or there are some specialized databases which are targeting for a particular organism or serving a particular research community. So this is the homepage of NCBI, that is National Center for Biotechnology Information. It was established by NIH, National Institute of Health. Now the function of NCBI is to develop certain computational tools or softwares for analysis of biological data, as well as to establish databases for the storage of variety of biological data. So this is one of the database established by NCBI, PubMed. Now, PubMed or Medline. Medline cover field of medicine, nursing, dentistry, preclinical sciences, etc. And it contains citations of approximately 5,200 worldwide journals in 37 languages and 60 languages for older journals also. So it contains over 20 million citations since 1948. So if in future, whenever you want to search for the particular research article, you should go to PubMed website or PubMed homepage and search for the articles. So this is how the search page of PubMed look like. You simply have to type in your query or keywords here in this box and click on the search button, and you will get all the research articles which are published in the digital world uh, as a result, and you can simply then access the article through here. Okay, so when you click on the particular article, you can actually read the abstract, or if the text is free, you can also click on this link and you can access that text article. What are biological databases? Now, in the biological databases, the information of or the data of nucleotide sequences, protein structure, it is stored. Now, based on their content, biological databases can be roughly divided into three categories. That is primary databases, secondary databases, and specialized databases. So primary databases contain original biological data. So if a researcher have determined the DNA sequence or protein sequence, they can directly submit that data to the primary databases, because primary databases accept data which is unprocessed, which has not been transformed into any kind of information. So they archive raw sequence or structural data submitted by scientific community. Examples are GenBank, which is a nucleotide sequence database, and PDB, that is protein data bank. It is a protein structural database. Then there are secondary databases. Now they contain computationally processed or manually curated information based on original information from primary database. So here in the secondary database, nobody can directly submit their own data. There are curators for these secondary databases. They pick up the data information from primary databases. They computationally process it or manually process it, and then they submit that information into secondary database. So the information or data that is present in the secondary databases, it is more accurate, we can say, as well as it is annotated. Then specialized databases are those that cater particular research interest. For example, suppose a group of scientists working on HIV or group of scientists working on Drosophila melanogaster. Then there are specialized databases for them, like HIV sequence database, which will serve the purpose of researchers who are working in the field of AIDS. Or Flybase, it is a database of all the sequence and protein structures of Drosophila melanogaster. So it will serve the particular research interest in the field of uh, Drosophila melanogaster.
In case of primary databases, there are three types. One is for nucleic acid sequences, other is for protein sequences, and third one is for protein structures. So the most prominent databases for nucleic acid sequences, they are GenBank, DDBJ, and EMBL. Then for protein sequences, we can use SwissProt, Tremble, that is translation of EMBL, then PIR, that is Protein Information Resource, and UniProt. Fortunately for structural databases, there is only one type of primary databases present, that is PDB, that is called as Protein Data Bank.
So what are primary databases? As I mentioned, the primary databases contain raw sequence or structural data, and it is the original biological data which may be unprocessed. Now there are three major public sequence databases available, that is GenBank, the EMBL, European Molecular Biology Laboratory database, and the DNA Data Bank of Japan. These three are freely available on internet. A small number of sequences, especially those published in 1980s, were entered manually from the published literature by database management staff. Presently, the sequence submission is precondition for publication, as I mentioned earlier also. All these three databases, they collaborate with each other and they exchange the new data entry on the daily basis under the collaboration name International Nucleotide Sequence Database Collaboration. Although the three databases all contain same set of raw data, each of individual databases has slightly different kind of format to represent the data.
Under international sequence database collaboration, these three databases share the information with each other on the daily basis. Like, suppose you submit the sequence to GenBank, EMBL, or DDBJ. They will share that sequence information with rest of the two databases on the same day. So on the next day, you will find the same sequence information present in all the three databases. So you can search the GenBank with the search engine called as Entrées. Then you can search the EBI, that is European Bioinformatics Institute databases, by using Sequence Retrieval System (SRS) search engine, and you can search the DDBJ by JetEntry. So these are the search engines which are available for searching all these three resources, that is NCBI, EBI, and CIB of Japan.
Fortunately for three-dimensional protein structure databases, there is only one option, that is PDB, Protein Data Bank. Now this PDB contain three-dimensional coordinates, which are also called as the Cartesian coordinates of proteins as well as nucleotides, which are determined by X-ray crystallography or NMR. So all the protein structures which have been determined by X-ray crystallography or NMR, or which are predicted by computational methods, they all have been submitted into this single database called PDB. Then secondary databases. Sequence annotation information in primary databases is very minimal. Now, annotation information means the description of the sequence. Like suppose you have a gene sequence and you don't have any kind of description with that, you will have no idea that what is the function of this gene or what kind of protein this gene codes for. So sequence annotation means when you have a sequence along with its all description. For example, there is a gene sequence whose location on the chromosome is known, whose protein product is known, whose function is known. That is called as sequence annotation information. So in secondary databases, all the sequences are annotated, means their information is present, their description is present. To turn the raw sequence information into more sophisticated biological knowledge, much post processing of the sequence information is needed. Now, prominent example of secondary database is UniProt KB. It provides detailed sequence annotation that includes structure of protein, function of protein, protein family assignment, etc., etc. And these entries are called as annotated entries. The annotation of each entry is carefully curated by human expert and thus it is of good quality. In case of protein annotation, it include protein function, domain structure, catalytic site, cofactor binding, post-translational modifications, metabolic pathway information, disease association, and similarity with other.
These are some examples of secondary databases. Now, in the secondary databases, we cannot directly submit the information. Usually the curators of the secondary database, they gather the information from the primary databases, they manually or computationally process that information and they submit the information into the secondary databases. So the examples are ProCite, PFAM, Prodome, Prints, and Blocks. Now, in these secondary databases of the protein sequences are analyzed by multiple sequence alignment methods, and then they are classified on the basis of protein family, protein domains, and all that protein family and domain information is stored in this secondary databases of the protein.
Sequences. In case of, let's say, take example of a gene sequence. What kind of annotation of a gene sequence may have? It may have a open reading frame information. It may have a functional site information. It may have a structure and function information. Like in this example, you can see this is the gene sequence, but we do not get any insight into this sequence by just reading the nucleotides. But when you analyze that sequence and you identify that this is the promoter, this is the transcription factor binding sites, this is the open reading frame, this is a stop site, then that sequence can be called as annotated sequence like this. Here you can see that this is the promoter site indicated in the red color. This is transcription factor binding site. Then this is ribosome binding site. And these shown in the blue color are the open reading frames. So this is called as sequence annotation, in which the whole information of that sequence is provided. And that is the work of secondary databases. It provide you annotated sequence data. Then specialized databases. Specialized databases normally serve a specific research community or focus on the particular organism. Now, the content of these databases may be sequence or other type of information, and the sequence in this database may overlap with primary databases also, but may have new data submitted directly by authors. Example, Flybase, which is dedicated towards Drosophila melanogaster, Wormbase, it is for Caenorhabditis elegans, etc. Now, as I mentioned earlier, that there are three nucleotide sequence databases present which are used widely. One of them is GenBank. Now, GenBank is most complete collection of annotated nucleic acid sequence data for almost every organism. It content include DNA, mRNA, cDNA, express sequence tags, etc., etc. The resulting flat file contains three sections: header, feature, and sequence entry. So let us discuss one by one. The header section describe the origin of sequence, its identification of organism, its unique identifiers associated with the record. The top line of the header section is called as locus, which means the location of that sequence in the database. Then the identifier is followed by sequence length and molecule type. So this is followed by the three-letter code for gene bank division. There are 17 divisions for gene bank according to organism or maybe some other sort of classification they use. For example, PLN is a division for plant and algal sequences. PRI is for primate sequences. PCT is for bacterial sequences, etc. So there are 17 divisions in which the sequences are classified at NCBI GenBank. So this is how the sequence look like when you retrieve it from the NCBI GenBank. This is the header section. In the header section, you can see here the locus. Locus is actually the location of the sequence in the database. This is the accession number. This is the length of that sequence. Type of molecule, it is linear. The division is BCT, that is bacteria, and it was published on 15th June 2002. Then definition, that is the name of the sequence, light independent photochlorophyllite reductase subunit N, its accession number, its database source, its keywords. These are the keywords. Then references, in which research article it was published, etc. Then feature, if some annotation about that sequence is available, then it will be provided in the feature section. For example, source, it was isolated from Heliobacillus mobilis. The gene which code for that protein, it was BCHN gene. The protein is light independent photochlorophyll reductase subunit N. Its enzyme commission number, and finally the sequence. So this is the sequence of that amino acid. These are different types of biological databases that are available today. UniProt, ProCite, FASTA, Emboss, PDBJ, that is of Japan, etc. Major databases for nucleotides or nucleic acid, three major databases are there. One is EMBL, that's in Europe, GenBank, that's in USA, and DTBJ, that's in Japan. For protein, it is PIR, Protein Information Resource, MEPS, SwissProt, Tremble, NRL3D, and latest one, the most important one, that is UniProt KB. Then nucleotide databases like EMBL, Ensemble, Genome Server, GenomeMod, EMBL Align, Parasite, Mutation, IMGT, etc., etc. These are protein databases like SwissProt, Tremble, GOA, Protein Profile, etc., etc. And as I told you that all these databases, they are interlinked, they are correlated with each other. Means let's take an example of a genomic data, GenBank, DDBJ, EMBL. Now all they are cross-referenced with other databases like protein databases, disease databases, gene expression databases. Somehow you find a correlation in between all databases. And also that correlation is highlighted in the form of hyperlink so that you can click on redirected to the other cross-reference database. So these are 10 important bioinformatics databases and their URL address: GenBank, Ensemble, PubMed, PIR, SwissProt, Interpro, OMIM, Enzymes, PDB, and KEGG. This is the database which contain whole human genome sequence information. It's a Homo sapien genome view. You can see all the chromosomes and the whole genome sequence information of human, it is stored in this database. This is the PubMed I have mentioned already. This is the homepage of EMBL, that is European Molecular Biology Laboratory Nucleotide Sequence Database. You can again simply put your query here. You can type the name of the sequence and click on find. You will get the results accordingly. This is the protein data bank, again the primary database for protein structures. You simply have to type in your query here and click on the search button. You can access the protein structural information in this database. So this is how the three-dimensional structure of the protein look like, and all the information about that 3D structure is given here. All right, so that's all for now. Meet you in the next lecture. Thank you.